enzymatic receptors. So we already discussed about basics of enzymatic receptors. We discussed that these are the receptors which have two ends, extracellular and intracellular. On the extracellular phase, the drug binds, and on intracellular place there is usually presence of some enzyme. So when the drug binds outside, the enzyme is activated inside. So we will discuss little bit detail about these enzymatic receptors. Enzymatic receptors can be broadly of three types. One, the drugs which have intrinsic tyrosine kinase activity. Intrinsic tyrosine kinase activity, the meaning is when we give a drug, it binds on the extracellular end, and that will directly stimulate the tyrosine kinase enzyme which is present on the intracellular end. That means these have and receptors themselves have tyrosine kinase activity. No extra enzyme recruitment is required. So drug bind outside and the enzyme activated inside. And this type of intrinsic tyrosine kinase activity, it is present for the insulin. So insulin, remember we discussed pig hormones. Out of them, insulin only, it has itself tyrosine kinase activity. That means intrinsic tyrosine kinase activity. But on the other hand, there may be some enzymatic receptors which do not possess the enzymatic activity themselves. No intrinsic tyrosine kinase activity. Means these themselves do not contain the enzyme on the intracellular end. Rather, they will recruit some enzyme from the cell, within the cell. So the drug bind outside, it will phosphorylate some of the proteins present on the enzyme here, and that will recruit some tyrosine kinase from the cytoplasm, and that will activate the tyrosine kinase. Then the reaction occur. Means the enzyme itself do not possess the tyrosine kinase activity. So one example of these drugs which do not have tyrosine kinase activity, so they usually activate another enzyme called as Janus kinase, Janus kinase, which will recruit another protein known as STAT. So Janus kinase will recruit STAT, and that will result in finally enzymatic activity. So this JAK-STAT pathway, or the enzymes which do not contain the intrinsic activity, enzymatic activity, these are examples of these are rest of the hormones. That means among the pig hormones, insulin has intrinsic activity. So rest of the two, that means prolactin and growth hormone, they do not possess intrinsic activity. And all the cytokines, they can act by enzymatic receptors, but they do not possess intrinsic tyrosine kinase activity. Okay. Then third type of enzymatic receptor may be which do not contain the tyrosine kinase inside. So the drug bind outside, the enzyme which is activated inside is not tyrosine kinase, it is guanylate cyclase. Guanylate cyclase. So the drug may activate guanylate cyclase enzyme inside. Drug bind outside, thus guanylate cyclase is activated inside. So obviously that will increase the cyclic GMP, this enzyme. And the substances which act through guanylate cyclase, these are natriuretic peptides like ANP, BNP and CNP. These have enzymatic receptors which act through guanylate cyclase. Okay. So these are the details about enzymatic receptors. Clear?